Hello and welcome to another episode of Clean Talk. It is 3 p.m. on Wednesday, and that means we are coming to you live from the beautiful Seal Shield Studios here in downtown Orlando, Florida. Very excited today to have as my guest Clinton Beeland, founder and CEO of CJB Industries, including CJB Applied Technologies. Clinton, welcome to the show.
Hey, thanks, Brad. Appreciate it. I'm glad to be here.
Well, we appreciate you being on. What more can you tell us about CJB Industries and why you're here on Clean Talk today?
Sure, be happy to. CJB Companies, really a family of companies, CJB Industries, CJB Applied Technologies, and the newest, which we'll talk quite a bit about, Salvis. We're about 25 years old, have 140 great employees, really helped build the business from scratch to in March of next year, 25 years. So we're really excited about that. About 8 years ago, we founded CJB Applied Technologies for developing products. CJB Industries is actually a toll or a contract manufacturer of chemical products, predominantly used in the agricultural market. Applied Technologies then develops things for others as a contract and also does a few things on their own. And out of that came the niche which we have, and that's Salvis. Again, about 140 people, all kind of scientists, chemists, operators, depending on what we're doing in packaging and processing. So my wife and I are the owners. We are real excited about where we are today and what the future holds.
Well, fantastic. And yes, so in case they're wondering, CJB stands for?
It's Clinton and Gina. My wife's name is Jeana, Beeland. And oddly, when you start a business, you're not real sure what to name it. Those initials kind of looked good on a logo, so that's how we came up with it. It's not the sexiest of naming things, but it works well for us.
A great tribute to the founders. So you mentioned that CJB is really a player in the agriculture space. Can you tell us a little bit more about CJB and the kind of business you do there and how that led to the Salvis project?
Sure, absolutely. So CJB Industries has been doing agricultural products for, as I said, about 25 years. And so we make products for small businesses, medium-sized businesses, and multinationals. Many of the products that farmers use are produced by us. In that market, some years ago, we've noticed an opportunity in the market with dicamba, or one of the chemistries that's used in the field, and it had a low level of contaminant, which could harm a crop that was susceptible to the chemistry. One of our advisors suggested we look at how do we clean the tank for the farmer, or how do we look for that contaminant to make sure it's gone. And so we went up and visited Georgia Tech, asking if they could help us. And so from that came a new technology, and that's what Salvis kind of evolved from. And we'll talk more about, I think, the device and its evolution, but that was its start, and that was back in 2015 or 16.
Well, we are going to talk about Salvis. You mentioned that a lot of applications in the agriculture space, which is your specialty at CJB, but I get excited because of potential in the healthcare space. And as you know, this is an infection control podcast. What can you tell us about the Salvis detection technology, and describe the Salvis technology detection unit?
Absolutely. And what we did is came from the detection we looked at dicamba. And then, of course, in 2020, we all had this exciting thing called COVID evolve. When we began with detecting the chemistry, we understood that the technology had broader application. However, we were going to begin in the area that we knew the most, which was agriculture, production agriculture to begin with. And then we would expand into potentially medical or healthcare applications, as well as other industries. When COVID came about in 2020, we made a call back to Georgia Tech to our chief scientist, said, hey, we can detect COVID. And we would kind of ran that same, it's kind of ESP. Suddenly she had the same thought just prior to the call. And so we devised a plan to look at both antibodies and the virus. And that sped up our process to some degree. Of course, as you know, the supply chain's been real interesting, so it also costs us more. And I think everybody doesn't always see the impact of one versus the other. It created an opportunity, but it's an opportunity at a cost. And so that's how we arrived at more in the healthcare market earlier than we had anticipated.
So COVID, again, a catalyst for innovation and for application in that market. We're going to talk more about the Salvis detection system and how it works, but you mentioned that you developed this with some assistance from Georgia Tech. I know you've worked with some other research organizations as well in the development of this project. Can you talk a little bit about that and what it's been like to work with research schools to help develop a private solution?
Sure. It's an interesting, even though we have about 140 employees, it's still it's a challenge. It's different. You're working with large universities. Georgia Tech is world renowned. Fortunately, I'm a student of Georgia Tech or went there many moons ago. So, and had a lot of connections. And so that was the first place we turned to. We're in Southern Georgia. It just was a natural fit. So they had the root technology, developed it many, many years ago, and then we've turned and applied it into the market. But in addition to that, we've also found it helpful to work with Emory University, also world renowned. There's a partnership between Emory and Georgia Tech, both in Atlanta. One is very much in the engineering and science field, and Emory is very much in healthcare and other disciplines. So it's a nice marriage of the two institutions. And then and for us as a private developer, it's a good fit for us. We've also worked with the local university, Valdosta State University, and some of their laboratories that they have, and as well as an agreement with the local group as well, or University of Georgia. So we aren't, even though I went to Georgia Tech, we are not biased in any way, and we look for the resources that these universities have, and they have great resources, and they are amenable to working with private industries because that gets it to the marketplace.
So able to leverage some of the research institutes to help develop the product, but it is your product, right? Have you filed patents on this technology?
So we have a couple of things. We have license agreements with Georgia Tech for the underlying technology, as well as anything that they specifically did. And then we have patents ourselves. We have close to 18 that we filed. Many of them a few are provisional. Most have been non-provisional filings. So we're well on our way with that. And I'm sure out of this, as you continue to develop technology, you continue to find new and novel things that can be either patented or kept as a trade secret, depending on the application.
Well, I see the prototype unit sitting on your desk there beside you. I know we're going to talk more about the technology, but for some context and for our viewers, what can you detect with the Salvis detection system? What kind of pathogens?
Sure. It's a unique platform. It's capable of both pathogen detection, viruses, bacterium, some components of those, and chemical. So it has a dual platform, and it's handheld, small, deployable in the field. So it's a very unique technology in that respect. It's about having the receptor. So if you have something that'll receive what you're looking for, then we can depose it on the cartridge and then detect the analyte, or what the pathogen or chemistry of interest would be.
Okay, so you mentioned that it has to be on the applicator. What are we talking about here? Are we talking about liquids, air sampling, solid surfaces? What can we do with this?
So a lot of versatility. It has the ability to detect, or the fluid can be either liquid, of water of sorts, or it can be air. If it is a solid surface, then we can also wipe the surface, extract the wiping, and detect it in a fluid or an air sample. So there's a lot of versatility in that manner as well.
Now, you have to know what you're looking for, though, right? It's not just going to plug and play and tell you whatever pathogens in the air. Tell us a little bit more about that.
Right. There are people working on that kind of technology, and that, it's interesting. And you mentioned earlier what COVID has inspired. I mean, there's a lot of technologies coming out into the world. Some are early stages, may never get commercialized because they just don't have a pathway. And some can detect a lot of different things, I suppose, or at least theoretically. In our case, no, it's very targeted. It does have multiplex capabilities, so we can, under the current chip, as long as we don't need a reference channel, we can detect up to 4 different pathogens or chemistries. But you do have to know what you're looking for. So you can't just put a sample in there and say, what is it? We wish. That's something we've seen on Star Trek. And I think, those things evolve and we may get there. Again, there's interesting technologies that may take us in that place, but it's very specific to what we're looking for.
And let's talk about how it works. So the technology is laser interferometry, right?
Correct.
What is that?
I'm going to give it a little bit two answers. One, the basically we take a laser, we're going to split the beam, and one of them will become a reference beam and the other will be the analysis beam. And then we have a camera that picks it up. Probably from a layman perspective, it's a little like taking the flashlight when you're a kid and you produce shadows, and the camera picks up a shadow, and one doesn't have one and the other does. And through calculations and computer programming, it determines that pathogen is present or a chemical.
And using this technology in the Salvis detector, how precise, how accurate is this method?
It can get very low. It can get near molecular results. It's going to depend a bit on, again, what we're looking for and what the makeup of the sample solution is. But we get down to near molecular levels. For COVID, for instance, when we did COVID, and I'll use antibody in this instance, we've detected on our laboratory unit down to 1 to e to the 4th, which may not mean so much, but it's very small amounts of antibodies in the sample. In that case, it was blood sampling. When we speak of particularly medical terms, we speak of specificity and selectivity. And so our specificity for this small test was 92% and our selectivity was 100%. So you've got real accurate, and that's on our laboratory piece. We've got our device here that and this is an appearance prototype, but the actual prototype that runs the samples is just about to be complete for us to revalidate those tests on our actual commercial prototype.
Well, that's very interesting. And I'm going to take a pause here to let folks know, our audience know, in full disclosure, you and I have known each other for a long time. So we have met through our Vistage peer mentoring group, which is a group of CEOs that advise each other. You advise me, I advise you. Although I think, to be fair, you advise me a lot more than I advise you. I appreciate that. But I always find it interesting why someone who has a very successful business, such as yourself, gets inspired to take risks, right? To innovate something new. What made you decide that why not just keep doing what you're doing? You have a very successful business in the ag space with CJB. What makes you decide to roll the dice and go all in on something new, novel, and highly risky for development?
Boy, I'm going to get into philosophy or something. We're not here very long, right? The world's short, life's short. So we can choose to do that. And I choose to invent or to find something new. Now, risk is relative. We take calculated risk. So how much can we afford to do? What can we afford to do? And who do we need to participate to help elevate it? So it's interesting. And our business, even in the agricultural space, highly innovative. When you look at it, we have to feed the world. The world is growing. We have more intensive agriculture. How do we do that? So there's an inspiration. I think as a company and the people we have that work with us, it's what we want to do. We work so many hours in a day. It would get real boring, I think. Now, sometimes we like to be bored, but real boring if we kind of did the same thing over again. Now, there are things that need to be repeated over again, much like a diagnostic device. You want it to give you the same result every time. You want somebody to prove it and it's right every time. But we want to come up with ways to prevent that or ways to measure it. And so I think it's just a spirit of our business, and it's exciting, and that's what we enjoy doing.
Well, I have a great appreciation for innovators like you. We make the world go round and make it a more exciting place. And I certainly appreciate the development that you're doing specific to the Salvis pathogen detection system. Obviously, I can see tremendous applications in the healthcare space that we service. But beyond that, what are your expectations for this product, and what other market applications do you foresee?
Sure. You get into market strategy, and we have a platform technology that has application in a lot of different industries. As a company and the size of our company, to go and to target every one of those industries builds a massive corporation in and of itself. So we find it more effective to go out and find partners in those markets, and where we're producing the cartridge, we're producing the assay for them. We're providing the this form factor or other form factors depending on the application, and then they're delivering it to the field. There are some smaller markets where we may directly deliver, but we're going everywhere from the healthcare to animal health to the row crop or production ag. To industrial markets and water markets or water fields. So all of those have impact on the world, and all of them, particularly as we continue to grow as a world and more people, we're closer together. We have to have more intensive agriculture. We have to have more aquaculture, things of that to feed the world. We're going to introduce more pathogens and things of that nature, and also beneficials. So we're looking at how do we measure the beneficial things that are there to make sure they're at the right level, and how do we measure the threats that might come in that are going to interrupt those beneficials.
Well, so I understand pathogens. Explain beneficials. So give us some examples of beneficials that you'd measure.
So if you look at our a buzzword biome, right? So our biome in our bodies is made up of a lot of things. Even your saliva, there's over thousands of proteins and things in your mouth, and those are acting to digest food and stuff, right? So if you took an antibiotic, for instance, it's we're getting better. They're more targeted, but they're not always. They're going to kill some things that are healthy for your body. So how do we measure? What's your level of that very important beneficial that's in your body that you need? Did the antibiotic interrupt it too much where we need to take intervention and give you, in our case, I guess, probiotics or something of that nature? So those things are continuing to grow. And when we look at our agricultural sector and traditional chemistry versus biologicals, it's growing. And it's growing probably for the right reasons. I mean, we have to be in balance. The world, our bodies, our function, and we're a little off kilter. COVID hadn't helped, right? It made us a lot more off balance. But
that's for sure. And you mentioned the antibiotics, and I think there's a lot greater awareness now that people understand some of the risks. Really the miracle drug that has protected us so long, but the result, obviously, we're seeing the advent of superbugs that are the direct result of broad spectrum antibiotics. C. diff. Absolutely a result of the overuse of broad spectrum antibiotics. And as you said, we are getting better at that. I see the prototype system on your desk there. Is that something you can show our audience? And
I can pick it up again.
It works. Yeah,
it's an appearance prototype. The screen is touchscreen. The device weighs about 2.24 pounds. This was our first appearance prototype that I happen to have here. We've done some modifications to how it holds for a small hand, large hand, make sure it's effective. The cartridge, which we have a couple of different ones. One is actually this is actually a prototype, but the back of it does have a chip in it, which is the kind of the brains of the system. This also has an EEPROM in it, so it's a smart cartridge. And you'd place it into the device, and then whatever workflow we have for the particular analyte, then you would follow that. This is connected, so it has connectivity, Wi-Fi, USB, cellular. Also has a GPS capability. So there's high capability in this device to report out, to monitor if we're using a flow cell versus our single use cartridge. But even that would be able to locate where am I, what did it do, what kind of test did I perform, who was it on, and then that data goes up into up into the cloud, and then we can disperse that data depending on what kind of regulation. HIPAA laws cover a lot of this when you're in healthcare, so we have to make sure we're HIPAA compliant. But when you're outside of that law, it's still who owns the data, and there's some important things that we've done to make sure that that is kept appropriately. So small unit, you can see it in my hand. I have a pretty big hand, but we've placed it in my wife's hand, and other folks have smaller hand. Very comfortable. 2.25 pounds is its current weight. Though there are a lot of things we can do to bring that down, and the current form factor can be adjusted for certain applications.
So help me understand kind of the use case application workflow. You've got the cartridge. Now, the cartridge is pathogen specific?
Cartridge is pathogen specific or chemical specific, yes.
And then the cartridge is also where you put the sample. Is that how you analyze the sample on the cartridge? So you've got a pathogen or chemical specific cartridge. You put a sample on that cartridge, plug it into the device, and you're able, using this laser interferometry technology, to determine if there's a match between the cartridge and the sample. Is
right? To very low levels. The sample for the single-use cartridge is very small. It's about 50 microliters, which is about a droplet of blood's about that size. So I'll give you an idea. But then many samples, when you get into something like saliva, it's a very difficult thing to work with. They're thick or thin. You've got to deal with a lot of different things in them. They also have, as I mentioned, proteins. And in many cases, you're detecting a protein or a spike protein like you hear for COVID, right? What is that? Where is it? And so these other things can interfere with it. So you either have to create a solution or small sample prep prior to introducing it to the system. But most all, if not all, diagnostics today require some sort of preparation. The key is to make it simple. And can it handle what we would classify as dirty samples? How much other stuff can be in there, and it still give you an accurate result. And they're very specific to those samples, but highly accurate when you have that worked out, and highly specific depending on the receptor.
Now, you said earlier that you can test liquids, air, or even hard surfaces. Walk me through that workflow. I can understand how you can put a drop on the cartridge and identify a liquid sample. How would it work for air testing or hard surface testing? Do you have to take a swab, or what is the process?
Air sampling is actually direct introduction. So it would be through a flow. So instead of what you see on the end of this cartridge where it's confined, it actually has a tubular flow through the cartridge, and basically a pump pulling the air into the cartridge. A really small one, but a pump. And so then it's interrogating the air for the analyte of or the target that you're looking for. For a surface, you would actually have to wipe it with something, right? Because it doesn't magnify the surface, so you'd have to wipe it, or if it were a leaf or something like that you're looking for, you may digest it in something. So just depends, and then the wiping then would be digested into a solution. You could, I suppose theoretically, you could take some air across it and all, but that's not what our workflow would be right now. It'd be wipe, take that, either pour a liquid over the top of it that takes everything that you've wiped into a solution, take that solution, drop it into the cartridge.
Well, let's first of all, another question. So if it's pathogen or chemical specific on the cartridge, are you only detecting one specific target at a time, or can you make the cartridges for multiple detections?
So you can do it's multiplex. So the term multiplex means multiple targets. So the current design will do up to 4 targets. Now there are complexities to targets. Your buffers can be different. You've got to measure those, but that's its capability at the current state. Theoretically, you could make that as many as you would like, right? You line up multiple units, multiple things, pass it through. There are limits just physical limits. If you've got a 50 milliliter blood sample or microliter blood sample, when does it no longer become effective running through the microfluidic channels? So there will be some limits. 4 is certainly what we have in the current device.
So I'm very interested with the air application, and I'm trying to wrap my head around this. Let me see if I've got this right. So COVID's on all our minds. COVID has changed the way we think about a lot of things. You could have this Salvis detection device set up for COVID. It will detect COVID, right?
Mm-hmm.
And through airflow, you could have these devices located within a facility and be able to identify if there's COVID in the air. Is that right?
Theoretically, you could do that. We've not done any of those testing.
Okay.
You may have to concentrate the air depending on the level. So COVID's in the air. We know it's contagious from the air, from a saliva sample and things of that nature. The amount or the level that might be in any environment is variable. And so our limit of detection would determine whether that was a single pass through or whether you needed to concentrate that air before it ran through the unit.
Well, that was going to be my next question. So what determines if you have to concentrate the air? Like how much of a specific pathogen or chemical needs to be in the air to be detectable with the Salvis detection system?
So I'm only going to give you the example of the one that from COVID antibodies. We're down to 1 to the E to the 4th. I don't remember exactly the calculation to get there. That's a very low level, but that is at a higher concentration than air would typically be. So you would need to concentrate the air down to so many particles per cubic meter of air. And so you'd kind of determine where is that and then what can detect it at that level. And then how fast can you do it? When you get into something like PCR, it's having to run cycles and cycles. It can get very low, but it might take you, hours to get there because it's kind of replicating the cycles until it actually gets enough generated for it to detect it. Similarly, we would have to concentrate the air for this one if it's below our limit of detection. And at this point, we don't know what the low limit is. We have a theoretical idea, but we don't know exactly what it is yet.
So that's the air. You mentioned PCR testing, and that's something that now we've all become familiar with, right? We all know about rapid tests and PCR tests, and maybe you saw a previous episode of ours with David Johnson, who talked all about his experience running a COVID testing lab. Great stuff. Check out those previous episodes on cleantalk.tv. But is this an alternative to, for example, a PCR test? Can you spit on the detection cartridge and identify COVID or some other kind of pathogen that way? Is that an application?
It is an application using saliva as the sample medium. It doesn't do the same thing a PCR does. PCR replicates the RNA over cycles. However, it is close. We call it close to molecular. PCR is considered a molecular test. It's the gold standard, and it's what we would measure against when we're doing pathogen detection. At this moment, we're not at PCR, which could theoretically, I guess, be one COVID in a sample. But we're working to reduce that line of detection, and we're hoping that we get very low. So E to the 4th for our antibody at the moment. We have lower detection limits, picograms per milliliter, which is really tiny. That's parts per trillion of some chemistries. So the capability is there, but there is a place where the laser and the camera can't see anymore. So it does have a limit. We're just not certain where it is yet until we get to that exploration to say, hey, here's the definitive limit of the technology. And it'll be test by test. What is that level?
Well, the technology is developed out. You're obviously in the prototype phase. What can we expect? When do you foresee this product being commercialized, and when can we look forward to seeing the Salvis detection system in the marketplace?
I appreciate that. And it is moving very quickly, I guess, in COVID time. You have challenges in the market. The supply chain that we all hear about is certainly no less a challenge for us either. There are chips in this one, so that is has been a challenge, and it's definitely slowed down our development process. But we expect to be doing some commercial testing next year in 2022. So it's doing very well in that respect. When we finally get a channel partner directly in the market, I'd expect late 2022. There are some that we could go faster, but faster takes economics and purpose. So
it's all time and money, right?
It is time and money. Yeah.
Well, we look forward to seeing this technology in the field next year. Clinton, we do have a couple of questions from our audience. How quick does it read the results?
So for the antibody testing, for using our laboratory model, not the actual commercial prototype, which theoretically should be quicker, we can get a result for, say, a blood sample, no prep for that one, in less than 5 minutes. So it's very quick. We expect it to be faster on our device, but until we've proven it, I don't have a number to give you.
So Alyssa Gronko has asked another question. She says, with flu season here, it would be interesting if this could test for multiple viruses to rule out one versus the other. That's theoretically possible, right? That's
theoretically possible, yes.
Right.
So we want to do, so in any kind of evolution, you take one step at a time. So it's one molecule, one piece, one function. Otherwise, you're running parallel paths, which is fine, but it's back to time and money. But we certainly have that on our scope. Flu A and B, or coronavirus versus flu. Yes, theoretically possible. There are hurdles when you do multiplexing, but they're there.
And we're all experiencing this, right? And you get a runny nose, and all of a sudden, do I have COVID, right? So,
right.
So I appreciate that question, Alyssa. And we're looking forward to the technology being commercially available as soon as next year. Here's the big question: what does this cost?
That's always the question, right? And for any business, it's about market cost. What is the market? How does it fit into the market? What are the development costs for that specific assay, and how does it work? What we can say is we've run models for scalability. It's very scalable. The cost as we scale certainly comes down. However, what is the price and what is the application? So not 100% prepared to answer it because we don't have an absolute, this is the target we're going for. Here is the price for that. And we're working with our channel partners to set those numbers to make sure they're appropriate.
Spoken like a politician. We'll come back to that and nail you down on that at a future date. Clinton, I really appreciate you being on the show today. You've been watching Clean Talk live from Orlando. Our guest today has been Clinton Beeland, founder and president of CJB Industries. Thank you for joining Clean Talk. Join us every Wednesday at 3 p.m. for another live episode of Clean Talk. And until next time, remember, keep it clean.